All right, so here's a little better picture that shows you all 5 of our characters this week. These are all the white blood cells you're going to have to pick out. So of these 5, let's see, we'll flip over to page 22 in your packet, and you have them all here. You can remember them by never let monkeys eat bananas. Neutrophil, lymphocyte, eosinophil, and basophil. And so that's all of them if you forget all their names. One other thing to take into account about their names is they get their names based partially on their appearance. So for these granulocytes, and the granulocytes are the ones that have these distinct little, these have small specks here, these bigger granules in the cytoplasm, and the basophils have huge granules so you can see them really well. That's why they're called granulocytes. They're one major category. And then agranulocytes are the ones that don't have granules. But again, if you look at these granulocytes, you have neutrophil, eosinophil, and basophil. And all 3 of these names tell you something about their appearance. Neutrophil, the neutro part, means neutral. And so this actually tends to have both a pink and purple appearance. If you remember with the hematoxylin and eosine or H&E stain, which is what we use in the lab, they have a purple component that sticks to basic structures, and they have a pink component that sticks to more acidic structures. And so the neutrophil is kind of in the middle. It's both pink and purple, kind of blends in, so it gets this neutral name. So it's a neutrophil. Eosinophil likes the eosine component of the hematoxylin and eosine stain, and eosine is the red component. And so we saw this with acidophils in the pituitary as well. It's kind of a synonymous name. The acidophils liked acid. These eosinophils, you can kind of think of them as the acidophils of the white blood cells, but they call them eosinophil because they attract the acidic component of the stain, which means these granules specifically are really red, and you'll see a better picture in a little bit. But their key component here is they are going to be red, and that relates to the name. Basophils are basic, so they're going to be really purple, basically. They're kind of the opposite of the eosinophils in color only. So again, don't just blindly memorize the names. Just remember the names have a meaning, and they can help you not exchange them and help you remember what they more or less should look like. Then our agranulocytes, they have no granules, and there are only 2 of them, lymphocytes, monocytes, and we'll talk about those more in a second. All right, so first we're just going to work our way through all of these, starting with neutrophils. So neutrophils, number 1, they make up 40 to 70% of the leukocytes in your blood. You may see different percentages. I'm not going to be too picky about the percentages. Let's, I guess, stay with the numbers that we have in our packet of pain. So packet of pain says 40 to 70% for leukocytes. So they're the easiest ones to find. When you look through the microscope, chances are the first white blood cell you run into is going to be a neutrophil. So neutrophils, they go through phagocytosis, which is called cell eating. That's their main function. They're going to eat bacteria. That's all you really need to know for the function in lab. But for identifying them, their distinguishing feature is that they have between 2 and 5 lobes of the nucleus. And so here, a lobe, they're all, oops, sorry, go back to this slide. They're all they're all mononucleate cells, meaning they only actually have one nucleus. But the nucleus has multiple lobes, so this is a little bridge right here between this lobe, and this could even be a 3rd lobe potentially. Over here you clearly have 3 lobes, but neutrophils can have 5 lobes. Sometimes it appears as if they have 6 lobes, but the point is their distinguishing feature is the number of lobes in their nucleus, and they're going to be between 2 and 5, and normally they're going to be 3 or more actually. And so the rule I'm going to use for lab for neutrophils is that in any neutrophil you need to identify, there will be at least 3 lobes of the nucleus present, and as many as 5. And again, they're also going to have a neutral appearance, so they won't be especially red or especially purple or anything like that. But the distinguishing feature, the one that you can rely on regardless of the color or the brightness of the slide, is the number of nuclei. And again, there'll be 3 or more for this one. And you're going to run into these all over the place. Eosinophils are going to be a little bit harder to find. So these are these red ones. You can see it really nicely in this particular slide. You can see all these red granules. So again, they're the ones that like eosine, and that eosine is red. So they're going to be a little bit more red than those neutrophils. They're also going to have 2 lobes in the nuclei. You can't quite see the bridge in this picture, but it does exist. And so their distinguishing feature, the one that you can rely on 100%, is that they will have a bilobed, or they will have two lobes in the nucleus. No more, no less. And so, and they'll usually be nice and neat like these ones, rather than with the neutrophils, things get kind of messy and it's hard to pick out if they have three or four. With the eosinophils, you have two clear lobes of the nucleus, and you will have a more red complexion. But again, color is a hard one to totally rely on, so don't only rely on color to make your decision. Your distinguishing feature is going to be two lobes. Basophils, one more thing about eosinophils. Take a look again at that percentage. They are 1 to 4% of leukocytes, so these are going to be hard to find. You're going to be searching around, you're going to be looking at neutrophils, and you're going to think, oh, I think I've ran into a bunch of eosinophils. And you'll think you see like 5 of them. Chances are you're actually looking at neutrophils rather than eosinophils. Eosinophils are real rare, so you're going to have to search a while to find them. Their function for eosinophils are to kill parasitic worms. So you sushi lovers out there, or people that consume raw animal products, tend to have high eosinophil counts, and that's a good thing. You know, you can battle those parasites, but that's the main function and only thing you need to know for eosinophils. Basophils, these are the hardest ones to find. Many of you will never see it in lab. When I find one, we'll have everyone come over and look at them, but it's because there's very few of them. Their main function is histamine release, and so histamine is involved in the inflammatory process. So if anyone has a sinus infection right now or something like that, you actually could draw your own blood and look at it and would probably have a pretty good chance of finding basophils, because inflammation, they're a major part of inflammation. So if you have any inflammation going on, you're going to see high basophil counts. But in normal individuals with little to no inflammation, you're going to see less than 1% of these guys, so they're going to be hard to find. They're pretty small, tons of granules, and their distinguishing feature has nothing to do with the lobes of the nucleus, because you can't even see them. So their distinguishing feature is that you can't see cytoplasm, you can't see nuclei, they're just a sack of granules basically. And they're very, very coarse granules, so you can almost pick out where one granule is and where another one is, and they really do stand out. But it's going to be hard for you to find them. And they're almost always a very deep blue color too. All right, so lymphocytes. So we just went through the first three, the neutrophil, eosinophil, and basophil. Those were all the granulocytes. Now we have two more left, and these are agranulocytes. Lymphocytes, these are also really common. About 20 to 50% of all leukocytes are made of, or are lymphocytes. So you'll find these a lot too. They're quite small. They kind of look like basophils, but they don't have granules in them. And they do have visible cytoplasm that shows up as this blue crescent. And so if we flash back to basophil, you know, on this picture, sure, they look pretty different, but if you imagine looking through a microscope, being zoomed out a little bit further, and you see this small blue circle, and you see this small blue circle. If you don't notice the little crescent of cytoplasm, you could really convince yourself that this lymphocyte is a basophil. And so be aware that this is a common thing, the lymphocyte and basophil. On your picture, or on your sheet right now, draw a circle around both of those together, like link them together, and warn yourself, I'm likely to confuse these two, basophil and lymphocytes. The key to not confusing yourself is whenever you think you found a basophil, make sure you search long and hard for this narrow crescent of cytoplasm around it. Because if you look and you adjust the fine focus, maybe adjust the brightness a little bit, you'll get this very subtle crescent to appear. And then you'll say, oh, okay, that makes sense. It's a lymphocyte. It's not actually a basophil. All right, so the last agranulocyte is a monocyte. These guys are easy. They're gigantic. Their distinguishing feature is size. They have a monolobe nucleus. That doesn't mean it's circular. It can come in all different shapes, but it's one major lobe. And it has a ton of cytoplasm. And if you look at the size of this cell, they do vary in size, but they're always significantly larger than the red blood cells around them. And so if we go back to these guys, see this lymphocyte, it's about the same size as a red blood cell. Basophil, same thing. Eosinophil, it's a little bigger, same with neutrophils. But really, the monocyte's just gigantic. No granules, really, really large. Lots of clear cytoplasm with no granules in them. And their main function here is phagocytosis. So again, they're going to also be eating things. That's why they need to be so big. They can take in very, very large things. I forgot to mention the function of lymphocytes here. It says it on your sheet. Produce antibodies, destroy virally infected cells and cancer cells. There are actually a lot of subcategories of lymphocytes, and they're really, really important. You'll learn about those more in lecture. But function is production of antibodies, and they deal with viruses and cancer cells. All right, so that is it for the white blood cells. I'm going to kind of pause here. The one last category are platelets. And again, the platelets are not really cells. They're just fragments of cells, like here and here and here. And they're just little specks that you're going to see all over the place. They're obviously, well, not necessarily obviously, but people have heard of platelets before. They're involved in blood clotting. So if you have a low platelet count, you're probably going to have issues with blood clotting.